Hi, Bradley.
Hello, how are you?
Hi, fine, Bradley. How are you?
All right, doing pretty well.
I've been seeing a lot of research paper related GCN. I got some pretty clear idea on what is it about Devo Graph and all. I found some new methods also to implement. I want to show you. I was very excited about this meeting since 2 days.
Oh yeah, that would be great. Yeah, you told me you had some things you wanted to show.
Yeah. Actually, I was reading a couple of papers related to segmentation. I found out that DevoWorm was good for segmenting raw data, but there is an issue with it actually. When the cells were like so many cells, it won't segment properly. For that, there is a best method called QCANet. I wrote some slides. I'll show it to you.
Okay. Hello, Susan.
Hi.
Hey. Yeah, I look forward to it. Well, you can share your screen in a minute. I just wanted to make sure that we had people coming in. Looks like Susan's here now. So how are you, Susan?
I'm fine. You can hear me okay?
Yeah.
Oh, okay. Yeah. I have I did a PowerPoint that I could present as well, but doesn't have to be today. So
okay.
It's just about cell cytoskeleton. So
okay, that sounds great. Yeah. All right. Well, Sushmanth, do you want to share your screen?
Yeah. Yeah, you can see the screen, right?
Yeah.
This is actually like a paper called 3D Convolutional Neural Networks Based Segmentation. They are using some raw segmented fluorescence images for segmenting data. Actually, it is done mainly for mouse embryogenesis, like it is used for mouse cells. But they have implemented for it for like this also, for CL GANs also.
Okay.
After that, by comparing results with other models like CNN approach, convolutional neural segmentation, and UNet, QCNet was showing better results compared to these two of them. About this is QCNet, like it's a robust, accurate algorithm to acquire 3D position of cell, would help to reveal mechanism of embryogenesis. Like we need 3D fluorescence bioimages. Actually, this algorithm is completely based on image processing algorithms. They fail to some of the CNN fail to detect an image when the object doesn't fit into the pattern that algorithm can process. So algorithm was mainly based on heuristic image processing. I didn't know the meaning of this. I tried to search, but I forgot. Sorry.
What is it that you're asking
changes
about?
From high.
What were you asking about?
This one, the fluorescence images.
Oh, yeah.
I don't know what they are. I asked Mayuk actually about this thing. Like what is fluorescence images? He told like he never analyzed the data, but Epic data set was completely fluorescence. He told. Even Hari Krishna Pillai, last year student, was there. No, Bradley. I talked with him also about this thing. Like what is fluorescence images yesterday? He was like he don't have an idea. Can you just tell me like what is fluorescence images?
Sure. So it's a type of image that's they use ultraviolet light usually, and they use a marker that has a fluorescence in it. So it could be like an antibody stain that has like a fluorescence dye in it, or it could be like something that has a fluorescence marker. They usually use fluorescent elements from other animals that they put into a cell. And you can see if you put it under UV light, you can see these fluorescent markers, and these usually give some point of reference.
Okay. Before presenting that, I asked Mike that does a big data set was completely fluorescence. He told it's fluorescence images only. Then only I thought of proceeding to this paper. I'm sharing this paper also in the chat box. You can see it. Wait a minute. Yeah.
So a lot of times, if it's a black image with some bright dots and bright dots throughout, that's a fluorescence image.
Okay, and These are the in QCANet implementation, whatever the paper is completely based on. It has two image pre-processing things like NSN and NDN. And nuclear segmentation network just segments the nucleus inside it, and the NDN segments the what we call outside the perimeter of that. After post-processing, we will get a 3D segmentation like this. So, and these are implements, like these are the photos from paper only. These are implements for C elegans, like ground truth. Actually, they have trained the model. They have compared it with ground truth. We don't know ground truth, but they used some fuzzy open source platform to get the ground truths of them. And they have like implemented QCANet. Then they compared and they have trained the model. The QCANet was able to solve problems of nucleus fusion in test data, which is not solved by 3D unit, and that of missing nucleus detection, which was not solved by 3D mask or CNN, like CNN implementation also. QCANet was robustly performing instant segmentation for images acquired for different imaging conditions. Here I mentioned like using Fiji open source platform, they have found the ground truth of fluorescence images at 11 time points. And this is QCANet consists of this what I told NSN and NDN. And the model architecture, like for NSN and NDN, they have mentioned in the paper like NSN hyperparameters were determined by Bayesian optimization. It is completely coding part which we can handle. And NDN, this is part of NDN actually. I hope once you read through the paper, you will get a pretty clear idea. I mean, it is good. It is better than DevoWorm when the cell nucleus are very large. Like not large, so many cell nucleus are there means it really segments well. When it is four cells are there, it's quite good. DevoWorm is performing well. But when these cells are increasing up till 35 around that, this wasn't performing well, whatever DevoWorm platform is there. It was completely built on ResNet 18 actually. And this affects on Devo Graph also actually, because Devo Graph is using DevoWorm pipeline to get the segmentation of cell nucleuses, right?
Yeah.
Centroids of that. So maybe we can implement that. I was like halfway there. I'm editing some code parts there. Maybe I will show Mayuk this thing. He said like, okay, you can just pass it on, get some raw segment data. If it's possible, we can integrate into DevoWorm this QCANet implementation.
Yeah. This looks good.
So maybe it will be used in Devo Graph also. I talked with Jiang Li, asked him, can we use segment raw data? Then he told like he want to just develop Devo Graph perfectly because segmentation works well with the DevoWorm. But actually, segmentation doesn't working well when there are so many cells. So that's the reason. And I was going through I have made a list. I will probably share by tomorrow about all graph neural networks. I was seeing some Stanford classes regarding GNN, and have seen through this paper also, like microscopy videos implementation, graph neural network, which was based to Devo Graph.
Yeah.
It is quite good, but we need some things to get cleared. I asked like Jiang Li, can we have a meet or something like that? He haven't responded. He was kind of busy. Then I asked Mayuk. Mayuk was so busy.
Yeah.
Actually, Bradly, he mentioned about you.
Okay.
He actually released a paper 2 days recently. It was the 5th best research paper in the DAR.ai community. Even it was competing with Google some of the research papers. It was like weekly's best paper ever released. I asked him like, how do you get this out? While I was working with Bradly, I learned so many things. Those I have implemented here. He was telling like that.
Oh, that's great. Yeah.
Actually, yeah. Yeah, that paper was like big blast. It was completely some lawmaker to ChatGPT. Actually, we know ChatGPT works fine, but it is giving correct predictions or not. It is not like defined. So Mayuk Dev completely worked on it. He cleared some like code type of thing which judges like ChatGPT what it was seeing in an image or in a text, what it is focusing on. It is completely about neural network interpretability, transformers, etc.
Oh wow! Okay. Yeah, I think I have a bookmark. I'll have to go read it in depth. So
okay. Okay, and I have seen another one also, lineage population method. There is a framework called ACE-Tree actually. I think you know this one also, Bradley. I haven't read it completely, but I'm sharing it here in the chat.
All right.
It is for a model implementation of lineage population. This is a tool actually. There is a repo and code also there. There is an open source platform to extract these lineage populations of an which is parent cell or daughter cell like that. Right now, I didn't got any clear idea about GNN, but this week I will totally analyze it if it's possible. I will next week I can show some best output regarding GNN. This was the best thing we can implement for segmenting raw data actually.
Yeah.
For the first stage one, these methods.
Yeah. So the things you posted in the chat, you posted the thing on ACE-Tree, and then that's the one that you just posted.
And then another segmentation.
The 3D convolutional neural networks. Yeah. So the nuclear segmentation network, is this like, I guess I don't understand what they're doing. Could you bring that slide back up?
And they are actually segmenting whatever data we had, like fluorescence images are there, right, into 3D structure. From those 3D structure, we can extract these centroids actually, centroids of every cell, which is used in, which can be used in Devo Graph stage 1 pipeline. Actually, Devo Graph has two stages. One was to extract centroids of each cell when we segment it. After that, we extract lineage population model through that, which is the parent cell, what was happening. But by this platform, we can get best accurate algorithm, better than DevoWorm, to get segmented raw data and centroids actually. And by this lineage population open source network, we can clearly get a CSV file to pass it to stage 2 to get graph neural network.
So it creates a 3D structure first, and then it segments it.
First creates.
Okay.
Yeah. And that's it. I have gone through stage 1 this week. What are the best methods we can implement to get accurate algorithm for Devo Graph? I found these two are the better methods, till now. We need to just integrate them, which fits in Devo Graph pipeline. We need to do a lot of work around it to fit into Devo Graph pipeline. And can I ask one question? What is future plan of this DevoWorm or Devo Graph? What do you want to make? Any 3D structure you want to make, 4D structure, which depends on time.
Yeah.
Cell dividing, like that, or do you want to just create a graph neural network?
Well,
that's the end of it.
The idea is to have this thing that you can implement on a data set. You can take out a set of centroids. You can build a network, and then so it has 2 paths. You can use one to extract centroids from some data, and then use those for some sort of analysis. Or 2, you could build a network or graph embedding, and then use that for different things. So we have, we have these 2 paths, and this is where Devo Graph comes in, because that's the other path. The first path is mainly just taking a data set, segmenting the cells, having some centroids, and definitely we're interested in the 4D perspective. So, the 3D perspective, of course, you have to have that in place. But over time, we want to see, because a lot of the data, it's essential to have things in time.
Yeah, have you tried to make 4D graphs on a 2D piece of paper?
Yeah,
I have seen this thing. We want, your idea was the same thing, right? I have this thing in my mind that we need to create a 4D structure which changes according to the time. When the embryogenesis happens, the cell divides, right? At that time, we need to create some type of thing we can accurately see where this point, cell division was happening. You just want to track the cells out. This is the main idea, right?
Yeah.
Yeah, cool. I get it.
Yeah, you can, I don't know if this generalizes to 4 dimensions, but an old trick is to make, for 3 dimensions, is to make 2 views at about 6 degrees apart in 3 dimensions, and then view them with each through a different eye.
Okay.
Okay, so
Yeah.
you see it in 3 dimensions, even though it's two-dimensional images. I don't know if that will generalize at all to 4 dimensions.
Actually, can we use depth maps for this? Do you know this thing called depth map in deep learning? They generally make a 2D image to a 4D structure according to time. We can implement this thing on like image analysis for this, whatever we want to create. Depth maps actually can be used for this thing. We can use graph neural network.
That's a change in the brightness, right?
Yeah.
Actually,
I know like we can change from 2D structure to 3D structure by using depth maps. We can estimate some by using maps. How can we create each cell is changing? We can use depth maps for that.
Okay.
This is all works.
Let me give you another hint. Maybe if you made time a color.
Okay.
Okay. So you could use a sliding color scale.
Okay.
So you could have your depth. You maybe add it to a depth map, but that might be a way to represent it.
Yeah.
Okay.
Thanks, Bradly. Those are all my doubts. I need to see graph neural network actually. I've seen Gian Levi, what paper he was implementing. That paper was pretty good. There are some loopholes in it. I will mention in next week. I need to read it completely to get a better view of that paper. We can make it more. But I'm not getting what is this topological data analysis. Actually telling whatever will be there, I generally go to YouTube. I watch the video. I go for an implementation. But whatever GNN part is there is only one best videos for that. That is Stanford lectures about GNN.
Right.
And about topological data analysis, there is no video, like there is no good information they are providing. Even we need to read some book to get a clear idea on that.
Yeah.
I was trying to.
Okay. Let me give you another hint. If you take any 3-dimensional structure and you rotate it, and you watch a movie of it rotating, it looks 3-dimensional. Your brain can do that.
Yeah.
Not require 2 pictures, just a rotating object. So to get 4 dimensions, you could have a rotating object that is also growing.
According to the time. The 4th dimension will be time, I think.
That's time, and you make a movie, so the movie becomes the time.
Okay, time-lapse video. Like a time-lapse video, it will differentiate each.
Yeah, so you keep it rotating to get the 3rd dimension, but then you make a movie to get the 4th dimension.
Okay. Yeah.
Okay.
I got it.
That might work because that growing. It's like taking a movie of a growing tree.
Yeah.
Okay.
A time-lapse video, I think. Like
you could
it
do
will be rotating.
Tree growing. If the tree is rotating while you're doing the time lapse, you could then see it in the 4th dimension.
Okay. Yeah.
Okay.
Understood.
Yeah.
Bradly, can you just help me out with this topological data analysis? Actually, you shared one video in channel, Devo Graph and DevoWorm, about topological data analysis.
Yeah.
If you have any other resources, please share to me.
Okay, yeah, I will. I have some, but I have to go dig them up. I don't have them with me right now. But yeah, I mean, there's some. I don't know if people are doing like videos. Like graph neural networks has a pretty high bar of sort of understanding, which is hard to get, but that's something that, so I know that Jia Hong worked on this last summer and he came up with this, this paper that he liked and he implemented it and refactored it. And then, I don't know what the next step is there. I mean, definitely needs improvement, but it's, it's like there, probably better ways to do it, I guess. And then the topological data analysis would be like from the graph neural network, but that's something we can talk about later.
Okay. Cool. I got it.
Oh, okay. Yeah. Well, yeah. Thanks for that. That was good.
Okay. I mean, we can improve the stage 1 process actually for the Devo, like in Devo Graph by using these 2 methods.
Yeah.
We need to see stage 2 also. I will give you an update within a week. Like I'm like at edge of that. Like what will be the best method? I was thinking to have a meet with Jiang Li, but yeah, I think he was busy, might be. So and now this things comes, I will see.
Yeah. Well, thanks. Now I'd like to give a quick update on a few things that we've talked about in previous meetings, and I want to go over, so first of all, some news on a manuscript that we've been working on, and then secondly, revisiting a conference that was attended over the holidays. So the first thing is good news on the front of manuscript acceptance. Our submission to the Royal Society Interface Focus was accepted. So we have, see if they have the featured submissions page here. This is the page for the special issue that we're contributing to. And this is our paper, Embodied Cognitive Morphogenesis as a Route to Intelligent Systems. This is myself, Richard Gordon, and Jesse Parent. And this paper was accepted last week. So this will be published in Interface Focus in a special issue on making and breaking symmetries in mind and life. So this is the Royal Society Interface Focus. This is the journal. It focuses on special issues on different interdisciplinary topics. So I look forward to that being published. And it's available right now on the SciArchive. And the abstract is here, but we're looking forward to getting a formally published study very soon. The second thing is following up on the MEMoDevo symposium, and this was held in Paris over I think it was last month in December. And I went to this. I attended this conference for the group. This was put on by the Pasteur Institute. It was put on by a group of people in Europe who work on different aspects of mathematical biology, mathematical neuroscience. So this was the MEMoDevo Symposium, Mechanics, Morphogenesis, Development, and Evolution. So there were a host of topics we covered in one of our previous meetings. This is a network of the attendees. So the attendees have different interests. And if you zoom in on one of these nodes, you see that they have different interest areas denoted by the tags. And so you can see that there's this bipartite network here of different attendees and their interests. So I'm trying to find DevoWorm. I don't see it right here, but we're kind of in the middle of the two groups. I don't know where. Oh, here we are, DevoWorm group. So this was registered as DevoWorm group. Here are the tags, the interest tags, and you can see we're kind of on the edge of this network. Not quite connecting to this network. This network is filled with people interested in comparative biology, machine learning, microbiology. This group is, I think, more neuroscience oriented, self-organization, information theory, network neuroscience, and so forth. So that's kind of an interesting way to visualize the attendees and their interests. This is a blog post on the node, which is run by the Company of Biologists, which is a developmental biology blog. This is hosted by the journal Development. And I was going to write a blog post on this conference and some of the things that I saw that were interesting, but someone else already did this, so I'm going to defer to them for right now and go over their blog post. And I may publish a bit about some of the things that I found interesting at that conference later, but for now, we'll just go through this post. So this is by Romain Lever, who attended the conference in person. There was actually an in-person part of the conference in Paris, and then a virtual part of the conference online. So this title is To Conference or to Unconference, a little report of the Institut Pasteur QBio Symposium MEMoDevo, and that's Mechanics, Morphogenesis, Development, and Evolution in its experimental format. So this is the poster from the conference. As you can see, I can't make it any bigger, but as you can see, it's very nice. It was December 12th and 13th of 22, and there were a host of people on different topics ranging from standard developmental biology to mathematical biology to engineering and other areas as well. So the diversity and complexity of shape in uni and multicellular organisms has long been a source of fascination and interrogation. Since the seminal work of Darcy Thompson, the study of the emergence of shape, so-called morphogenesis, has been strongly influenced by the concept of emergence, whereby complex pattern and shape can be explained by relatively simple mechanical and mathematical laws. So if you go to Darcy Wentworth Thompson's book on growth and form, he talks about different mathematical models for looking at animal morphology, looking at seashells, looking at other organisms with very regular looking phenotypes. And I mean that in the sense that there are, there's a high degree of modularity, a high degree of patterning, and you can actually analyze these through different mathematical tools, different mathematical functions, and different types of mathematical transforms that he applies. One of the most famous images he has is of a fish that he takes and he puts on a measurement grid, and then he takes that measurement grid and warps it to match that phenotype, the landmarks on the phenotype, like different places like the tip of the snout, the tip of the tail, and so forth. And then he warps the grid again to fit other fishes. So you can look at variation between different fish species by continually warping this grid and then registering the mathematical transform that results in. So it gives us some indication of changes in shape over evolution. And you can do the same thing in development. So he introduced a number of tools that the book was a bit ahead of its time because it didn't really, the computer techniques weren't there yet. But now that we have high-powered computers, of course, we can do these types of analyses much more easily. So this is kind of the inspiration for this workshop. In parallel, the rapid progress of biochemical characterization of the regulators of cell signaling has opened the possibility to compare developmental programs and dissect the molecular basis of evolution. So we can take these types of mathematical transforms that describe differences in shape, but we can also look at the biochemistry of these changes. And we know from MEMoDevo that these changes are due to changes in, say, the promoter of certain genes, or the changes in gene expression over the course of development, and it results in size changes, shape changes, shape transformations, and so forth. So we want to be able to merge mechanics and mathematical laws. We also want to merge the molecular aspects of morphogenesis, and so that's what this workshop was about. And there were a number of speakers from different disciplines here. The discussions and talks that were presented here covered a large range of approaches: mathematics, fluid mechanics, soft matter physics, genetics, evolution, developmental biology, epistemology, and so on. And so a lot of the things we talk about in this group, and it was a very, very interesting set of talks. There were a lot of open questions that were raised that weren't really answered, but the point is, of course, in one of these types of events is to raise a lot of questions. So it also showcased the diverse set of organisms from mammals, birds, reptiles, insects, choanoflagellates, plants, algae, yeasts, and bacteria. So just a wide range of organisms, from single-cell organisms to multi-cell organisms that exhibit very complex behaviors. So this is a really interesting set of talks that sort of covered a lot of territory in terms of these kind of changes in morphology over development and evolution, different modes of development, different areas of the tree of life, and so forth. So so the morning sessions were an unconference, and they were live in Paris. And as being a virtual attendee, I didn't get to attend these, but this was interesting because it really kind of allowed people to interact. So I missed this part of it, and it is probably pretty productive, I imagined. I wasn't there, and that's where a lot of these open questions may or may not have been answered So you can see that they had the whiteboard, they were making notes, they were, kind of brainstorming things. And so that's something that we could replicate in this group in terms of our own interests. So they had a roundtable. The first roundtable addressed constraints on the evolution of shape, and how to reveal them experimentally. So this is this idea of taking what Darcy Thompson talked about and looking at that from a more modern perspective, evolutionary and developmental constraints on shape. For example, what kinds of shapes are possible given certain conditions, evolutionary conditions, developmental conditions, and so forth. And then how to reveal them experimentally. So how to do experiments that intervene in that process to show some of these possibilities or show how these things are constrained. So how can an organ evolve from one shape into another? Are there limitations regarding shape innovation? Those are two questions that are related, because we know that, like in terms of like the Drosophila wing, that you can have shape innovation, you can have changes in the shape of the wing, but you can't necessarily have novel shapes for wings. Sometimes there are developmental mutations that modify the shape of the wing, but there are certain shapes that can't result in a functional phenotype, and there are certain shapes that can't be derived from what is there available to the organism. Exploring the distribution of organ shape in the morphospace. So they talk about the space of possible morphological configurations. Using intra and interspecific variability can reveal such constraints by looking at the non-occupied zones in the morphospace. So our organisms occupy this morphospace. We see some of these variations, some of these combinations that are more common than others, and some of them you never observe in nature. And so those non-occupied zones, as it were, are the areas we're really interested in understanding exactly what those constraints are and why they're the way they are. So the cause of these unoccupied areas can either be selective pressure or the funneling of evolutionary change by developmental constraints. So this is a very interesting topic. This is something we might return to in talking about sort of natural selection and development. And this tells you a lot about like the possibilities of what evolution can produce, what it can't produce from a certain starting point. The second discussion was centered on the emergence of multicellularity. So this is where you have single cells, and then they divide and they start to live in colonies, and then they start to live in even more closely associated groups that are multicellular groups. And so there's a lot of work on yeasts where you have clusters of yeast cells that form these multicellular aggregates. And so you can look at multicellularity as this sort of transition from a single cell to a multi-cell state of living. But in between there are some interesting structures that have that emerge. And to understand this transition, you have to understand some of those intermediate states and what it means to be multicellular, to live in the aggregated state. So by comparing the mode of multicellularity, clonal aggregative, the signals, conditions triggering aggregation or dissociation, which means to blow them apart and make them live separately, and the components that can structure and organize the aggregates, adhesion, contractility, the matrix. So all these things play a role in forming these aggregates, and we talk about them with respect to cell, more changes in cell morphology, and even some of the tensegrity stuff we'll talk about today. A very complex picture emerges with all sorts of combinations of strategies found in nature, outlining again the diversity of evolutionary paths leading to multicellularity. So we prepared this table where it kind of shows some of these taxa that they talked about that were featured at this conference, and some of the states of living. So you have unicellular, clonal multicellularity, aggregative multicellularity, sexual reproduction, symbiosis, and so forth. And then in these different groups, these taxonomic groups, you can see whether they exist in that state or not. So this is a nice way to analyze this type of diversity and to see these non-occupied zones. Okay, then there were the afternoon hybrid sessions, and those are the ones I attended virtually, and those were also held in Paris. Very interesting talks here. This was a more classic set of talks where they focused on different topics, and they ranged across a lot of different areas. So the first one was a historical overview of the evolution of approaches used to understand development. And this focused on mechanical constraints and shape and how people have done experiments to demonstrate those things and to bring that into the modern era. Then there were a number of talks on the cephalic furrow in Drosophila embryos. So the cephalic furrow is this structure that moves across the embryo and starts to give it some sort of shape, starts to allow for folding and other types of morphological transformations. And so understanding that as maybe just a mechanical phenomenon, or understanding that as something that interacts with gene expression and is caused by some sort of developmental program, is what they were kind of getting at in some of these talks. But we also had talks on plants. We had talks on petunia petals and sepals. We had talks on how mutants can be used to disentangle some of these different issues. So, for example, the furrow, is it controlled by genes or is it controlled by mechanical processes? The same thing with plant growth and development. What kinds of genes are involved, and if you knock them down, what are the effects? There are also other types of quantitative studies on different types of mechanisms. So mechanisms such as embryo compaction in mammals, how these types of mechanisms can be interpreted and understood. This talk here, there were a number of talks on topological transitions and how those things are analyzed and understood. So there were some studies of organoids, there were some studies in epithelial cells that allowed us to look at some of the changes in shape and the transformations in shape and how those can be understood. There was another talk by Jose Bico, who actually works with these inflatable shapes. So the idea is that you can build these models. They're inflatable models. You fill them with air, they inflate, and they take on a certain shape. And the idea is to mimic these morphological transformations using a very simple physical model that doesn't have any genes in it, doesn't have any of the biological structure we usually have in biological systems, but they have the shape, the conformation of shape, and they can, constitute the soft active material that we can play around with and see, what the constraints on shape are when they're under a certain stiffness, when they have a certain set of constraints applied to them. So it's very interesting work. There was more generally, there was a focus on soft materials or biological systems as soft materials. And of course, biological systems contain soft materials, but there's this idea of soft materials and living matter that we talk about in the meetings that they emphasized in this set of talks. So there, you can look at local differences in growth. In the inflatable shape model, you can use inflation to mimic growth. In soft active materials, you can mimic changes in growth, changes in shape by different sort of techniques. And so you can do this in culture, and tells you something about what these shapes are and how they make their changes. We also look at insects, looking at different gastrulation in different fly species. So identifying genes that explain differences in morphological changes between species, but also within the common ancestor that resulted in the different species. So he's looking specifically at the mode of mesoderm invagination, cell elongation, and the requirement for cephalic furrow formation. And then finally, there was this talk for the first day where they were looking at differential mechanical properties of tissues that explain the morphogenesis of the jellyfish canal network. So this is a network within a morphology that is explained by different types of mechanical theories, such as cracking theory. So you can look at the jellyfish canal network as a mechanical structure, as well as a biological structure. And so with more of the same, the next day there were some other talks on fluid mechanics, applying fluid mechanics to chick gastrulation, looking at different craniofacial shape evolution patterns across vertebrates, Darwin's, finches, bats, and crocodiles. There was a diversity of different types of changes that resulted in these different morphologies, understanding that morphospace and how those changes happened. Also looking at different swimming strategies and collective behaviors in Volvox, Gonium, and Calmiodomonas. These are all single-cell organisms, so they're very different from our context of like flies and mammals and other types of vertebrates and invertebrates. And so this just provided a nice contrast to some of those systems. Then we had a set of computational and theoretical considerations on the concept of autonomy and some of the core principles that play a role in autonomy and how that relates to these issues of growth and development. And then they talked about more physical models and applying those to viscoelastic deformation and other types of morphological problems, such as a maze growth against a physical obstacle. So looking at how growth, not just development, proceeds, and some of these physical models that might come into play and explain them. And then, of course, in yeast, we talked about the long-term experimental evolution experiment of Will Ratcliffe, where he's evolved yeast over many, many generations. And he's been able to evolve these aggregates of yeast cells and how they form these proto-multicellular colonies and other types of structures. So it's very interesting. There's also some very interesting physics work that they've been doing with that, biophysics, in the sense of, how do you describe these multicellular aggregates and their function? And then finally, Stuart Newman provided some interesting ideas about the concept of dynamic pattern modules, integrating gene regulatory networks into models of physical and spatial constraints in development and how those things relate to evolution. So it's very interesting stuff going on there. Like I said, again, I'd be very interested in exploring some of these themes further. There were some questions I had about some of these topics, and it's probably grist for another set of discussions in another meeting. So yeah, that's great. So Susan, you had a thing that you were going to talk about.
Sure. I have a new house guest. You can
I can see
her there.
Oh, at least it's not on your keyboard.
Now, she eats my papers. I print them out so I can sit and read them nicely, and she likes to play with them.
Oh, she digests them before you do.
Yes. Well, I have a new excuse for my homework. The cat ate it. Literally.
Yeah, the cat ate my homework.
Yes. Anyway, yeah, I have a PowerPoint I'm making. It's too long for today, but I can show you the cell biology part.
Yeah, it'd be good.
Okay, so I just have to figure out how to share my screen. There. Okay. Anyways, I wanted to see what you thought of this. So I've made this. Here's pictures of some epithelial cells, and they included the contractibility with actin and myosin. These are the stress fibers. Then they've got actin in a tissue on how it's connected.
Okay.
And they've got fermentin intermediate filaments. Fermentin, is it? I don't know. But they've got the microtubules.
Yeah, is this for presentation somewhere?
Yes.
Yes. Okay. You might emphasize that that one that's labeled cortical actin architecture, emphasize that that's at the apical surface of the cells.
Okay. Top view.
Yeah, top view. Top view.
Yeah, so this is a top view of actin, intermediate filaments, and microtubules.
Now wait, what's the view of microtubules? Is that top view or beneath the top?
Well, it just said that this is the apical view, that an image of microtubules.
Okay, but it didn't say where the microtubules are.
They're green.
Yeah, but it didn't say where they are.
Yeah, no, it didn't say how deep.
Didn't say how deep. Okay.
No,
do you remember Beth Besides paper where she found apical microtubules?
Yes. Yeah.
Okay.
I think it depends on the cell.
I don't know if you can get an image of those because she did it before the fluorescence techniques were developed.
Oh, okay. Well, anyway, the actin is in red and the microtubules are in green. And
yeah, I'm just saying you can't get the vertical component out of the picture.
This is looking down on it, so.
Yeah, I know. So
but we don't have a depth. I know.
You don't have that.
Anyways, so here the actin is in red, and here it is in green. Different dyes.
Okay.
And then the next one, this is showing where the actin and the myosin is in a T cell. So they tend to form a ring around the outside surface.
Is that an isolated cell, not an epithelium?
It's a T cell, so it's blood cell.
I've never seen T cells. Is it a flat cell?
They're amoeboid, so they change shape.
So why should it have a ring, or is it a whole surface?
It doesn't show it well. This is a slice. But basically,
okay.
So the actin and myosin is surrounding the outside of the ball, or the amoeboid.
Okay. Gotcha.
Yeah, yes, I need to say that somewhere in here. This is even more confusing. There you go. This is microtubules and actin mixed, and I've got way too many words here. The actin, I believe, is in red, and the microtubules are in this blue color. So they associate with each other. This is not the best image, but it was interesting because they've got actin associating with tubulin, at least in this image.
What cell?
Pardon? I don't know. I don't think this is in a cell.
Oh.
Actually, but I have to go back to this paper. So I put it in here. Like, this is sort of not quite finished. And then another top view of cells. And if you have a close-up, you can see where the attachments are. They kind of have corners. So this is like the model of the cell I was trying to make, where it has 6 sides and attachments at the corners. It kind of looks like this.
Some of them are pentagons.
Yeah, some of them are squares, some of them are pentagons, and it shifts.
Yeah.
Yeah, so this would be only like one point in time.
What is at the corners?
Actomyosin complex.
Oh, so the stain is the same along the edges as the corners?
No, there's this red. Yeah, I knew somebody was going to ask this. There's a red dye along the sides, and there's the green dye at the corners.
Oh.
So I'm assuming that the actomyosin complex is labeled green. I need more detail on that slide.
Okay.
But yeah, the myosin and actin just kind of act like a muscle, and they pull on the actin to pre-stress it, which you need in a cell or it'll just collapse.
Again, this is all of the apical surface.
Yes, so that's all. Was all the apical surface. Now this is a side view. I don't have a good side view as an actual real image.
Okay. Okay, Susan, I'll ask you a stupid question.
Okay.
Why doesn't the same thing happen at the base level?
Because there isn't this actomyosin ring.
No, why don't you get a ring at the base level?
And why don't you get a base here?
Yeah.
Oh, because this is an epithelial cell, and they have a basement membrane, and they like to attach to a basement membrane, and when they do this, then they don't do that. I don't know why.
You don't know why. No. Okay.
No.
Okay, I throw the question out. Why? Well, how does the asymmetry happen?
Why is there apical basal asymmetry in an epithelial cell?
Yes.
That's the question.
Throw it on as a question. I don't know the answer either. So.
Yeah, well, they tend to attach to this ring of actomyosin around here with cadherin in between here, and then the basal part of this is attached
Okay.
by integrins. So you have cadherins.
So is there a difference between cadherin or junctions and stuff at the bottom in its attachment properties to actin and myosin?
Okay, I'm not sure the integrin complex, but it has some. It just has attachment abilities, I believe, and the extracellular matrix kind of determines the stiffness of that attachment. So if the extracellular matrix is stiff, then the attachment is stiffer. It's very dependent on the extracellular matrix in epithelial cells.
And by the way, for axolotl, the proportions are wrong. In other words, at neural plate stage, it's about 25 microns across and about 50 to 100 microns vertically.
Yeah. Okay. What's the ratio?
You better look in my old papers or something. I think it's about 25 to 100.
Okay, yeah, because these are just short.
Yeah,
like these are like liver cells or something. They're boxed.
You've shown these as squares, but in the axolotl they'll be long rectangles.
Well, you can have long rectangles, and you can have these boxy things, and then you can have the really flat ones.
Oh, okay.
Like the flat ones are the skin cells.
Okay.
And they have a similar attachments.
Uh-huh.
Anyway, I have a picture of that somewhere, but not in these slides.
It's making you talk longer.
Maybe I need to be making notes here.
Yeah, okay. Okay. Look, it's before the presentation and a hard typing hole.
Right, yeah, well this is very rough. This is sort of okay, I need different shapes. And more explanation. Anyways, so they've got cell-to-cell junctions here. They've gone over it in fairly great detail. Oops. So you can have different junctions. So adherens junctions, desmosomes, hemidesmosomes, focal adhesions.
Which junction allows transmission of ions?
The they don't have them in this. Gap junctions. Oh, there they are.
Yep, yes.
Gap junctions.
Gap junctions, okay.
Yes, and gap junctions are not attached to the cytoskeleton directly. Like they're not attached to the actins or the intermediate filaments directly.
Okay.
They're just attached. They just kind of make a hole through the cell membrane, and you can pass proteins and, well, I think you can pass proteins. I don't know. It's a small pore. You can definitely put ions across that, and gap junctions occur in places like the heart.
Okay, which kind of junction is involved in axolotl neural plate?
In neural plate?
In other words, the ones for which we found the cell state splitters.
Okay. I haven't thought about that, but my guess is there's some tight junctions and some anchoring junctions there. There'll be,
Okay.
they have these X's for tight junctions to keep things safe because they're an epithelial cell. And then they'd have the cadherins attaching to the actomyosin ring in here. And they haven't included the microtubules, so they're missing something. This thing.
Okay. The microtubes attached to the junctions or not?
Do they attach to the junctions? I think they're just in here, and the actin sometimes attaches to the microtubules. They're just a part of this complex. That's my reading on it, but I took a whole mini course on, what was it? Was it edX or the other one? I don't know. There's a mini course on cell cytoskeleton, and these actin filaments have an interesting branching that they do, so they can hold each other up. So this is a branching network in here.
Okay. Can I make a suggestion? You know that old paper that Wayne and I did? It was 84 or something, where we gave many different configurations for cell state splitters. Do you remember that?
Yeah.
Okay. Can you modernize that? Bring it up to date in terms of all these junctions and whatnot.
Oh, okay.
Because we have a whole bunch of different configurations.
I can take a look.
For instance, there were microtubule rings that parallel the actin rings in some configurations.
Yeah. I assume it all depends on the cell and the.
Oh yeah, they were different. They were from different kinds of cells. The question is, can you update that information?
I'll look at it because I've certainly taken that course, and it's interesting if you push on your cell with a high force, you get more actomyosin happening or developing.
Okay.
And it has a different branching. It has a more shallow branch.
We gave references for each hypothesized cell state splitter configuration. So it would be possible to take the papers that we cited and do a forward search on them.
Okay. Yeah.
Okay. That might tell you whether anybody followed it up and gave more detail. Because we certainly did not have the information about connections and junctions.
Yeah, well, it's interesting. And my thought is that, you know how you had the microtubules held with the elastic bands out to stiffen it? You might be able to do that with the actin too, because it is a it has branches. So it's stiffening itself in some ways.
Okay. Is there an intermediate filament ring that parallels the actin ring?
Let's see. They had, I think this is, go back to the first picture. Of course, this is where they have the intermediate filaments, and that's around the nucleus in this case. But if I go down here further, I'll just continue here. So this, I think this is a better diagram because it shows it more in 3D. And here are your intermediate filaments.
Okay. Do you remember Chris Martin's work where he found intermediate filaments in a ring paralleling, I think it was slightly below and paralleling the actin ring?
Okay. So there's, where is it? Stable adherence junctions. So intermediate filaments are the blue. Actin is a red. There's definitely an actin ring there, and then microtubules. But I still think this is maybe missing something. I'm not sure. I'm interested in this because this is a soft surface.
Yeah.
And
really?
A tight junction and stable adherence junctions along the apical surface.
It looks like intestinal villi.
Yes, it does. It definitely does. And then they've got one here that's on a stiff surface, and they show the integrins here, twice as many attached to the extracellular matrix along here. And they have the actin fibers, the stiff actin fibers are along the basal part of it.
Or the apical side is turned upside down.
Yes, and then they see these are weak and unstable, but my thought is that if all of this is stiff, this is going to be more unstable. This is going to naturally break more.
Maybe.
Just because of the mechanical nature of it. But anyway, this is the confusing cell biology I've been looking at. And I have a stack of about 100 papers, and I need to read through them. And it's not clear.
Okay.
You think that they can make a stress strain curve? Why, no. They took the stress and they divide it by the strain and use that as a variable. I'm going, why?
Okay. Well, no sympathy. On another subject, I just collected 700 papers.
Oh.
I go through.
I had a question about the soft and stiff surfaces. The last two slides.
The surfaces?
Yeah. So it looks like you have a network of actin and microtubules, but you also have the surface, the thing that it's embedded in. So that's basically transferring forces into the network, or what's the nature of that material that it's embedded in? Is that play a role in the stiffness, or is it just the configuration?
It plays a role in the stiffness, yes.
Yeah. And so it's just the geometry, or is it the sort of the nature of the material?
Yeah, you need to know the stiffness of the ECM, extracellular matrix, or the substrate that the cells are on.
Okay.
It's very important. Okay. So I did state that here. You need to know the pre-stress, like how the myosin is stretching the actin, and you need to know the extracellular matrix. And it's also good to know where the actin ring is in a cell. So
one other factor, Susan, you have to know whether the surface is hydrophobic or hydrophilic.
Oh, okay.
Okay.
Yeah. Well, they usually make them hydrophilic.
Yeah, but have you looked at Ingber's study where he made little squares that were hydrophobic and hydrophilic, and the cell about the size of the cell? And the cells became very differently on the two different size squares. It wasn't an epithelium; they were individual cells.
Yeah. Okay. Well, I can look at that. Here they've just changed the stiffness of the basal membrane. So this is, instead of the extracellular matrix, they're attaching them to things like glass and gel. And they pre-measured the substrate or the extracellular matrix substitute.
Yeah, but again, you have to control for hydrophobicity because glass is typically hydrophilic unless you coat it with something.
Well, I think they did. They were careful about it.
Oh, okay.
Yeah, that's why I like this article, and it's an older one, 2006. And I like that they had a graph, so at least they had elasticity along one line, and it's a differentiation markers along the other. So the blue is nerve tissue, and you can see it occurs when the elasticity is less. And then you have, this is IOD, what is that, muscle?
Muscle, yeah.
Muscle in green, and so it's in the middle, it's kind of the middle elasticity. And then you have bone, and this is bone here at the higher end, and I maybe should have included it. I have a graph showing all the tissues in the body and how they, what their elasticity is, and it lines up perfectly with this graph.
Okay, they use the same kind of cells for all three cases?
Yes, mesenchyme cells.
Mesenchyme cells. Okay.
I don't know, I looked for the species, and there's no species said listed. So, this is how I'm going to mystery species.
The universal animal.
Yeah.
It's annoying.
It's a giant mouse.
Yeah.
It's either a giant mouse or human. Maybe it's human, and they didn't want to say why. But anyway, yeah, no.
Maybe it's human fetal tissue.
yeah, controversial human fetal tissue. That would do it.
Yeah.
I don't know. Anyway, it was 2006, and I didn't know this existed before, so I was in awe. So it's been included here.
Do a follow-up on that paper.
Yeah, I should. Yeah, I need to do a forward search.
Yeah.
I haven't done that. See, this is full of holes, this presentation. And anyway, so this is two ways of measuring things. One is with beads, and the other one is with micropipette aspiration. And they do get sort of similar graphs, but they're slightly off. This is pre-stress, and this is cortical stiffness. So they're similar. So I don't know if they're comparing apples to oranges, but they think they're comparing different types of apples. So there we go.
Okay. Just one comment there. The use of the word tense cells would suggest to some people you're doing a psychoanalysis of cells.
Okay.
Elastic.
How about we just do that? There we go.
Differ cells, yeah.
I like that better.
Oh, okay. Yeah, okay, fine. That's not psychoanalysis.
Yeah, okay, and I need to put, these are beads and this is micropipette. I need to put that in there. Beads, micropipette. I'm making notes. And that's slide 14.
Okay, Susan,
Yeah,
are you familiar with the old experiments where you put an oil on water and it spreads out and you measure the thickness of the oil layer? That would be from high school, maybe?
I sort of know about it.
Okay, it was used to measure the thickness of the layers of oil. Now, if you did that with epithelial cells, they'd be floating on water, so to speak, which would be a very low stiffness medium.
Yeah.
Okay, I just throw it out. I don't know if it's
Okay, well, the very low stiffness will turn into a blood cell.
Oh. How do you get a triangle?
Well, that's what they say. They turn into blood cells. If they're on fluid, they turn into blood cells. And if they're on compressed or pressurized fluid and being under pressure, then that does something else.
Okay, so hold on a second. I'll get a copy of the screen. Can you send me that paper?
The 2006 one here, this one?
The one with the triangle.
Oh, the triangle. Yeah, this is neat. I like this because you don't have to move anything. You just put your cells down on a triangle and.
Oh, you put them on the triangle. Oh.
Yes, and then the triangle tips are at a higher tension, and you can see where your mesoderm develops and where it doesn't.
Well, it's still like that because I'm working on triangular cells.
Oh, right.
Oh, I see. Okay. Yeah, they're stiff. They have a interesting pressure that occurs at the tips.
Right.
Okay, so.
Okay. Yeah, so I'd appreciate a copy of that. Whatever page that triangle picture came from.
And that's okay. All right.
Thank you.
Anyway, I like this. This is measuring, I believe, tension between the cells, and the rest of these have been the substrate measurement. And then this is again looking at integrins, and if you have a mutant that doesn't produce integrins or the fibronectin for whatever reason, you have problems. So in neural tube closure, this is a very sensitive part of embryo development. And this is looking at the tail, or the start of the tail, I believe, or maybe it's the whole thing. But this particular picture is from the tail closing. Anyway, even the shape of the neural tube changes if you don't have the integrins holding it to the extracellular matrix. So this is wild type, so this is the way it's supposed to look, and this is the mutant without the integrins attaching. And you can see there's a difference in the cells along the line. So
does that go to birth or no?
I believe it does, but I'm not sure because this is E18. It's almost ready to be born, and this looks distinctly different from that.
Yeah.
And there's the spinal bifida right there, and it's got a tail defect.
Oh.
But the tail doesn't grow.
Huh.
So Anyway, that's it's a problem.
So the top is mouse and the bottom is human.
No, this is a mouse. I can tell you this much. All of this is a mouse.
Okay.
At least I know that. Oh well, and so that's kind of the end of the cell biology, and then I get into stress strain curves. Why can't biologists do this with some of these lovely experiments?
Because they don't study any physics.
Well, at any rate, this is how much it stretches along the x-axis and how much force is being created on the y-axis.
Yeah.
And this is a standard in the industry.
Cracking up Sushmanth.
And some people have measured stress strain in tissue. So, and I think this is a bone. Anyway, tissue always has this toe. So it's sort of a J shape.
It's not toe tissue.
Yeah, they have a toe. It has a toe and then a straight middle part. And they call it a J, but it's sort of J shaped.
Sort of.
Yes. And so
Lazy.
some more examples of what they would consider a J shape here in muscle. And this is in brain. So it really does have this type of curve. Uh-oh, I'm getting an echo.
Okay, this is external stress.
Yes, this is stress applied externally. Yes.
Okay, does it show hysteresis or does it come back to the curve?
I think the brain might have shown hysteresis. There is sometimes hysteresis and sometimes there isn't, depending on the tissue.
Okay.
And if these, the brain in this picture, I know it's dead tissue, and it will be different from live tissue. So when I'm going to be doing some experiments, I really want to do experiments on live tissue. That's what actually works in a living system.
You can microwave it first.
And measure the difference between live and dead tissue. Experiment number 1.
Yes, you too can come up with a definition of life.
Yeah, there we go. And then there's this. If you it depends on the tissue how long you can apply force to it and how much force you can apply to it. So the time is important in here, and the amount of force you can apply to it is important. And a quick force is different than a slow-moving force. So there's all of that you have to keep in mind when measuring things. So yeah, so this is kind of me measuring tissue. Oh, I forgot one. Alive or dead. There we go. So these some of the things you have to keep in mind.
Okay, dentists presumably stay in the alive region.
So the what?
Dentists with their braces and things like that, removing teeth.
Yeah. Yes. So they want these relaxation curves to occur in your mouth.
Okay. Comment. This is a general property of viscoelasticity.
Yes, all of this, especially the relaxation and creep curves,
Yes.
measure the viscoelasticity.
Yes.
And some of the frequency response does as well. You just have to get the right frequency going, and quite often in, say, mammalian cells, it kind of, their natural frequency is the rhythm of their beating heart, whatever that is.
Oh, okay.
With people, it's 60 times a second, or a minute, sorry. And with birds and other small things, it's quite a bit faster.
Yeah. The hummingbird is the extreme.
How many what?
Look up hummingbird heart rate for the extreme.
Oh yes, yeah, that's right. Well, even mouse, like it's faster. They're smaller.
Don't go.
And so what is it? Oh, then there's different ways of modeling tissue. And the one I managed to get myself into, sort of by accident, was this, tensegrity.
Oh.
So there's a red intention.
Sushmanth, you can see a rotating structure. So it looks
there you go, yes. Rotating structure. And this is just from Wikipedia. They had it on there, a little GIF.
Oh, yeah.
Well, and then this is the tensegrity that I'm working with. It's a 6-sided one with 6 rods and how many strings? 3 times that many?
Okay.
18 strings.
Okay.
Yeah.
Yeah, so here it is, and there it is in MATLAB, and here it is in Console. And I can go back and say the top doesn't line up with the basal part of this, like it's got a twist to it. So in this one, this is the one that's straight up, and this is one that's twisted. And I had the straight up one made because you can't do a tissue with a twisted one. It was impossible. So in Console, because it's difficult to do anyway. I'm sure if you made one like this and then kind of twisted the whole structure, that might work. But
okay, Susan. Basically, a twisted structure is also chiral.
Yes, yeah.
Okay, so what you're saying is that epithelial tissues are never chiral.
Well, they can be because, or they can have a swirl in them, like the pattern of hair growth, for instance.
Oh, right.
Can be that, but these are malleable. I mean, they're essentially elastic. They're skin, so you could have a twist in them. That bears some more study. I mean, just looking at this just brings up issues.
Yeah, you need to scalp a few kids.
Yeah. What? Not mice? They always get experimented on. Anyway, so
mice have whorls. I know
mice
kids
have whorls.
Most kids have whorls.
Yeah. Oh, there's squirrels everywhere. Birds have whorls.
Oh, really? Okay.
Yeah. Anyway, this is just my experiment, and it seems to go over a really broad range of forces. Like, it's just quite amazing. This twisted structure stands up better than the straight one. So I did a couple experiments.
Okay, are there any observations of the structure of epithelia at the edge of a wound?
Yes, there are.
There are. Okay, it'd be interesting because there you have an imbalance.
Yeah. And I think the actomyosin, which is the string in this case, they just, and there becomes a gap. I think they join each other and then close the wound, or else you get something migrating in. I had an interesting paper about migrating cells.
Okay. Look, many years ago, I attended a lecture on making holes in the ears of rabbits.
Yeah, I think.
And I tried to do a calculation of the forces involved. And my conclusion was that something's going on strange because the hole closes up. If you make a hole in a rabbit ear, it eventually closes up. Whereas if you make a hole in a sheet of rubber, it gets bigger.
Yeah, well, they're alive, and I think they form an actomyosin ring that closes it. You got your actomyosin muscle-type movement, and I think it closes the gap. I'm not sure about that, but that's what I guess.
Wound healing of epithelia might be a source of information.
Yeah. Okay.
Especially if anyone's done the microscopy of it.
Right. Oh well. And anyway, so then I ran into this paper somebody had done with just the 3-strut.
Oh, right.
So it's kind of nice. It shows the movement here. It only goes until the strings touch or the rods touch. So, and when you apply a force, it winds down, so it becomes a different height. But they did this, they did it with elastic strings, and you get this sort of a curve. Oops, that doesn't belong there. There it goes. And if you do it with stiff strings, you get a J curve.
Oh.
And the J curve, this is more like what a cell is. It has a J curve. Like tissue has J curves. So I'm assuming that the Stephen Levin was right. Of course he is about this. And your actomyosin ring is quite stiff. And Dr. Ingber says it's like 100 gigapascals.
Okay.
Or 100 megapascals.
Oh, did he get?
Well,
he got past using strictly elastic tensegrities?
I think so, because he states that the actin bundles are very stiff.
Hey, Bradley, you can see the kind of hard time I give students.
Yeah.
Anyway, this is in compression and this is in tension.
Susan survived me somehow.
Yeah.
I don't mind your questions. They definitely add things. So he made a taller, he called it a slender model as well. So, and it has different J curves. And anyway, I need to hurry up with this. So tensegrities this is from Stephen Levin, they need to be J-shaped, and the tension elements need to be quite stiff. And this is a single blood cell. It's not in a tissue, and it has a more rounded shape. And this is a more rounded shape, but it's packed. Packed cells, and this is more inside of the body. So I spelled honeycomb wrong, good for me. So the epithelial tissues are more of a honeycomb shape, and the inner body tissues are more like these isohedrons, and are packed like bubbles. So I'm sure that comes from you, Dick. So anyway, so I love this thing that was on with
yes. Yeah, I didn't expect a demonstration for Sushmanth.
Yeah, well, there it is. Anyway, so any questions, or should I just turn off there?
Chuck, can you stop it rotating and show that it no longer looks 3D?
Well, I have these ones up here. Do they look 3D?
Yeah.
You have to show the
Yes, because you can see what's in front of what.
yeah. If you weren't an artist, you'd be going, yeah.
So these last two slides you showed. These last two slides you showed or the last two before the concluding slide were interesting because that gets into biological cells.
You can see it on the thumbnail. It's not rotating.
Oh, okay, yeah.
Oh, all right.
It looks flat.
Yeah.
It looks. The only reason why, where is it? This doesn't look flat to me, is because they have the lines here.
Right.
And this looks like it's on top of.
Brain fills it in somehow.
Yeah. So.
Yeah.
Anyway, that's kind of what I put together. It needs more explanations, obviously.
Oh yeah.
Okay.
Yeah, those last two slides, I wanted to go to those just to look them over a little bit more.
The last two slides.
Yeah,
here?
in the talk, like the ones the biological cells or whatever. Yeah, so that one, red blood cells. Yeah.
Way back here.
So that's interesting because that proposes some really explicit structures for a real cell. So the red blood cell, yeah.
This one?
No, the red blood cell one where you just were. It's way down at the end.
Okay.
Yeah, there.
Oh.
Yeah, that one.
So this.
Yeah, so that really shows some explicit claims about what they look like with a red blood cell, what the network should look like, and you have so this is from this paper, structural multidisciplinary optimization. So these.
Huh.
Yeah.
What are the molecules?
Pardon?
What are the blue molecules and the red molecules?
Well, the blue molecules are the stiff elements.
Oh no. Okay.
Which are, I don't know, you could say they're stiff elements. And the red ones are the cables.
Yeah, but what are they?
What are they? Yeah. I don't know whether they're stiffened microtubules or stiffened actin at the moment.
Yeah, because my old understanding of red blood cells is that they were structured by a network of some kind of other molecule, not a standard cytoskeleton molecule, that was adhered to the membrane.
Oh, okay. Well, they've lost their nucleus.
Yes, I know that.
Yeah. Okay. Well, just in general, that's why.
Yeah, there's a huge literature on trying to explain the shape of red blood cells.
Oh, okay.
Going back at least 40 years.
Okay. Yeah. No, I don't know if they explained exactly what the bars were.
Yeah. That's
physically.
Yeah. What molecule do they represent?
Yeah, I don't know.
That's why I asked, because the models I saw 40 years ago only involved the surface.
Oh, okay.
Yeah.
Well, I was trying to do tissue. I realize this is a tissue, but it's more like a single cell. So it's here for contrast because these are if you develop a mesenchyme cell on a fluid surface, you'll get something like this.
Okay. Yeah, it's nice because it describes the shape and it says why, what should we expect from that shape, and then you can go back and say, well, why is it structured like that, and what's maybe it isn't the same type of thing.
If you look at B, it looks like a sort of almost random conglomeration of stiff elements.
Yeah, and it could be. It doesn't have to be.
Okay, because that raises the question of dynamics. Suppose you made a structure like that of microtubules, but the individual microtubules were undergoing dynamic instability. You know what that is?
Yes. And that one might help it when it goes through one of those little micro vascular.
Bradley, you familiar with dynamic instability of microtubules?
Yeah, I've heard of it. Yeah.
Okay, the phenomenon is simple. The microtubule grows slowly, or it collapses very rapidly, about 10 times faster than it grows. So it's continually in a process of growing and collapsing.
That happens in the heart.
Okay, so if you made a tensegrity structure of stiff elements like these, but each one is not a fixed length, but it's growing and collapsing, does the overall structure remain intact or not?
It needs to fold to get through microcapillaries.
Oh yeah, of course. In fact, it becomes cup-shaped.
Yeah, so it has to.
That's a different phenomenon.
Well,
if you just look at the instability, if you look at the dynamic instability of microtubules, with this number of microtubules, do you still retain the structure, even though the individual microtubules are growing and collapsing rapidly?
Apparently, some of the kinesin and diin will stabilize a microtubule.
Well, that's stabilizing, but if they're not stabilized, does the overall structure nevertheless remain intact? Now that's a question that could be answered by taking a model like this and adding dynamic instability to it.
Yeah.
Okay. Yeah. And I think you'd come up with a critical number of microtubules at which the shape is retained.
Oh, okay. Yeah. So that's another study. Anybody want to study that?
Bradley whip it off my night.
Yeah. And then finally, that last slide. Yeah, number 39.
That's a question that's always bothered me. Most work on tensegrity assumes a fixed structure. But the dynamic instability of microtubules, of microfilaments, I don't know about intermediate filaments, questions whether or not, how many does it take to have a stable structure, even though the individual components are continually changing?
It is a good question.
Yeah, and I think it would be done by simulation now.
Yeah, it might be.
Okay.
Yeah, maybe console might be up to it. I'm not sure.
Yeah.
First, I have to get this full thing to work.
Yeah. Okay. Yeah.
Because
all you have to do is increase the length of the blue elements here, or decrease them individually.
Oh, okay. Here.
Presumably independently.
And then try it out. Yeah.
Try it out. Yeah.
Yeah. Well,
Now it gets a little more complicated because the rates are also dependent on what's called diffusion limitation. In other words, if they have to attract monomers of microtubules, tubulins, then they deplete them in the neighborhood of the growing end.
Well, if they've collapsed, the molecules are there.
If they collapse, they will produce them. They will produce free tubulin, and if they are extending, they will create a zone around them of depletion of the tubulin.
Oh, yeah, that's true.
Okay, so that makes the model a bit more complicated. But first order, I ignore that effect.
Okay. Well, I've got some of that trouble with. I'm having trouble with my model because you can go from 0, I think, to 1,000 newtons with this, and the thing still works. And I'm going, that's not right.
Okay. But again, these are static structures, so.
Yeah, but anyways, I've got to test. I've got to test my model, and I might just use this one.
Yeah.
Okay.
But,
so.
Yeah.
Anyway, I think it would make a great study in what is the stability of these structures versus time, hemodynamic instability of the components.
Yeah.
Okay, and that could be all done by simulation.
Instability. Yeah, because some of the structures are obviously not stable, and you can see that from my drawing up here that somebody did, and they say that some of this is not stable, in particular over here. So this is a weak, unstable adherence junction, and over here, microtubules, fast growth and fast breakdown.
Oh yeah,
so,
yeah.
So they're showing the instability in these diagrams.
Well, I think it's true also of actin, though.
Yeah. Well, they're saying that.
Okay. There's another question now. Are you familiar with treadmilling?
Yes.
Okay, good, because that's another thing that can affect things.
Yeah.
A microfilament or a microtubule can grow at one end and shrink at the other end. So there's
it looks like it moves, but it's not. It's just shrinking and growing.
Yeah. And that could also have an effect here.
Yeah, there's I have a paper on those two effects of the shrinking and growing, and
but not in the tensegrity context. Right?
Well, they're what keeps they think that the cell membrane is kept, the shape of it is kept by the actin filaments growing towards it.
Oh, okay.
That is from my course.
Okay.
Don't know if I can find the course. Let's see. It's hiding up. What is the name of the course? Yeah, I think this is it. Cell biology and the cytoskeleton with active matter. Let's see. Sorry, I'm not prepared for this. Oh, here it is. Cell biology, the cytoskeleton, and cell cycle, and it's edX. They have
okay.
I mean, it's interesting because the instructor actually is doing the research on this.
Okay, let me ask you a question on this picture here. They're claiming that you get a weak or unstable adherence junction far away from the ECM.
Yes.
Is there any way to estimate the force of pulling it apart?
I don't know. They pull on tissue and see if it breaks down. I'm not sure.
I don't know. You might want to look into that. See if there's anyone. Well, in this configuration, it would seem that there might be a net pull of two components of the adherence junction, so it pulls apart.
Well, there's not very much actin there.
Yeah, but it's not, it goes down to the actin stress fibers, some of them.
Yeah, there's some there.
Yeah, so at least in the cartoon.
Yeah.
So what I'm saying is it might be a tug of war.
Oh, okay. Well, is that what they call this? there was one called Tug of War.
Yeah.
Oh,
and I have-
and you could actually, if you could guesstimate that force, you could compare the guesstimate with, if somebody's done it, the actual force it takes to pull a junction apart.
yeah. Well, I'm sure there's some literature.
There might be.
Yeah. Well, again, I need to do a forward search on this. So this is just very rough, this presentation. It's,
Okay.
Yeah.
Pretty comprehensive.
Yeah, it
well,
is. Very
it's what I want to cover, but it's like not finished.
That's okay.
I just wanted to go to that last slide for a minute before we end. The one with the whole cell, inner cell tensegrity structure. Yeah, that one. So cells are often
Yeah.
truncated isohedrons. It's a space packing design, and then this honeycomb shape for the epithelial tissue. Okay.
Yeah. If I could spell this right, it would be good.
I mean, I wonder if different tissues have different sort of optimal packings, just because of the way that the cells are shaped and the function. It'd be kind of interesting to see. I
Yeah.
mean,
Let me ask a simple geometric question. Are you familiar with the close packing of spheres in 3 dimensions?
Are you asking me? You sent me a paper on that.
Anyone, just spheres.
I've heard of them. Yeah.
Okay, there are actually 2 different close packing arrangements.
Oh,
Okay, if you look at different layers, how the layers are stacked can be different depending on it can be different in 3 dimensions. So you get two entirely different kinds of 3-dimensional packings.
do you know what they're called?
No, I don't remember that. Look up 3-dimensional packings or something. I mean,
Yeah.
2 dimensions, it's unique, but if you stack the 2 dimensions to get your 3 dimensions, there are 2 different ways of stacking them. In fact, there might even be 3 different ways because there might be a random way of stacking them.
Well, certainly there could be a square cell in the middle of all of the other cells, and it would change configuration of.
No. I'm talking about perfect packings.
Oh, okay. Well, I'm sure that in a living cell or living tissue, they change.
They're imperfect. Okay, I'm just throwing it out as a consideration.
Okay. All right. Thank you for helping me with my presentation.
Yeah.
You mean giving you more work, huh?
Yeah. Well, you think you're bad. I get my professor yelling in my ear when I've done something silly.
Oh, I don't yell at people.
No, you just ask hard questions.
Okay.
Okay.
Right.
I'll stop sharing then.
Yeah,
Okay,
maybe
yeah.
share that 2 to 3 because I think there's also a random way of doing it.
Okay.
Okay. Well, I'll check out the pack.
Yeah, I think there may be a random way of packing different layers,
Yeah.
and they still end up with the same. Every sphere has 12 spheres around it.
Yeah.
If they're perfect. Okay, so these different packings all retain that property.
Okay.
Okay.
Yeah. I'm hoping that I'm just doing epithelial tissue right now. Probably not.
Yeah, 2-dimensional. Okay. Yeah. That's
well.
Bradly has to worry about 3-dimensional. So does
Yeah.
Sushmanth.
All right.